Hello, my name is Robin Hicks. My pronouns are he, him, and I'm the Dean of the Faculty of Graduate Studies at the University of Victoria. I would like to acknowledge and respect the Lekwungen peoples on whose traditional territory UVic stands, and the Songhees, Esquimalt, and Wasanich peoples whose historical relationship with the land continues to this day. It's my pleasure to introduce Mikayla Holmes, today's speaker in the Dean's Lunchtime Lecture Series. Mikayla completed her undergraduate degree in Math and Statistics at UVic in 2021 and is currently enrolled in a Master of Science in Statistics here. Mikayla's talk is titled Connectivity, Understanding Our Brain's Functional Organization. In this presentation, she'll explore how to use data from magnetic resonance imaging, or MRI, to better understand neural connectivity and how our brain at work compares to our brain at rest. Welcome, and over to you, Mikayla.
Thank you for the introduction. So today I'll be talking about background connectivity and understanding the brain's functional organization. So as you've already heard, my name is Mikayla Holmes. I am a master's student at the University of Victoria in the Mathematics and Statistics department. Now before I begin, I just would like to do a territorial acknowledgement. So I acknowledge and respect the Lekwungen-speaking peoples on whose traditional territory the University of Victoria stands, and the Songhees, Wasanich, and Esquimalt peoples whose traditional relations to this land continue to this day. So the general structure of my talk today is going to be a little bit of an appetite whetter, so my research problem, why it matters, who kind of cares about this, and then about half of the talk will be just general building blocks. So this is just foundational information that I think is really important to understand before I start to talk about the specific research that I've been doing and the sort of general project that all this research comes from. So what is the research goal? As I mentioned, our research goal is to understand the brain's background connectivity. So why do we care about this? There's kind of three main reasons that I think are important to point out. The first is that, in general, understanding our brain's background connectivity improves our understanding on how the brain regulates behavior. So that's all behaviors. It really plays a role in every single one of them. Secondly, we also know that understanding the background connectivity facilitates improvement on the building of brain maps. And so these brain maps can help experts in a number of ways. It can be for clinical assessments, it can be for intervention strategies. This helps medical professionals of all different kinds, neurological, psychological problems, and just different sort of behaviors like language, motor functions, etc. Now, the third reason is sort of the reason that motivates myself and other statisticians to research background connectivity, and that's because understanding background connectivity of the brain helps statisticians to improve all of the models that they are going to be building going forward, which will be applied to brain imaging data. So this would improve prediction power and accuracy in medical fields and psychological fields. So the first sort of concept I'm going to talk about with these building blocks is MRI technology. Structural MRI is probably the type that you've heard of the most. It's the most common type of MRI scanner. It is a non-invasive technology where magnetic fields and radio frequencies interact with the water molecules in our body. So the way this works, you can see by the picture here, is that the magnetic field in the MRI scanner basically aligns the protons in the water molecules. And then when a radio frequency pulse is sent through the MRI scanner, these protons sort of go back to a state of equilibrium in a way. This transition back to equilibrium varies depending on the type of tissues and organs that these protons are in. And so this is how we differentiate between potential tissues that have damage or that have a disease within them. And so that's why structural MRI scanners are typically used, or scans are typically done on soft tissues areas of the body. Now, the type of MRI technology that I will be talking about for the rest of the lecture is called functional MRI. Similar in concept to structural MRI, except for that functional MRI deals with how magnetic fields interact differently with oxygenated or deoxygenated blood. What we know is that increased blood flow occurs when neural activity increases. And so we want to measure where these neural activities are going on in the brain by measuring the increase in oxygenated blood. So the functional MRI scanner measures the strength of this blood flow variation across the brain which is then used to understand which regions function similarly when a subject is either engaging in a task or at rest. So these measurements, we call them blood oxygen level dependent. And I just want to reiterate here that this is a magnetic field interacting with blood, and oxygenated, deoxygenated blood, it no longer has to do with protons in water molecules. So moving forward, all of the observations and values that I refer to in my research are called the bold values, these blood oxygen level dependent values. So why do we want to use functional MRI scans? What is it for? So these scans allow us to understand this thing called functional connectivity. So what is this? Well, we know that different tasks require different functions of our brain. We also know that we can take images of the brain while the subject is completing a task or while they're at rest, and this gives us an understanding into which areas are activated during these tasks. Now, we say that when certain areas of the brain are activated simultaneously, that they are functionally connected. So this is the general idea about functional connectivity. So for example, here in this picture, we have, say, the yellow dots. They could be activated when a subject is wiggling their index finger on their left hand. So then we would say that these regions are functionally connected. So why do we care about this? Well, functional connectivity, as I said about background connectivity, helps us understand the role of our brain when we are behaving a certain way. So this can be helping medical experts to assess and plan intervention strategies when cognitive functions are abnormal. So if somebody has sustained an injury, stroke, heart attack, something like that, understanding the functional connectivity in the brain allows doctors to assess which regions are going to be impacted, or to figure out which regions were impacted by seeing those abnormal functions. This also allows doctors to monitor disease progression and the general impact on the brain function that a disease has. So functional connectivity, it's not the end-all be-all per se, but it is this umbrella term that we kind of use to describe everything going on in the brain that's measured by functional MRI scanners. I just want to point out this slide because we also refer to functional connectivity in another way, and it could be confusing. So essentially, when somebody is in the functional MRI scanner, they are either considered to be at rest or completing a task. We say that this sort of spans all states because resting state could be while you're asleep, it could be under anesthetic, or it could just be when somebody is doing nothing and resting. Now, completing a task, again, is the any sort of conscious engagement of an activity or sort of a question that they're trying to answer, a math problem, it could just be a physical movement. These are all different types of tasks. So from the resting state, what we get is resting state connectivity. So this makes sense, it's very concise, and the term is logical. However, with task states, we have this thing called, and I put task state or, but generally speaking, this is just called functional connectivity when we're referring to the stimulus that is task dependent. So the specific parts of the brain that are activated specifically to engage in the task that you've been asked to do, like complete a math problem. We also know, however, that there's other things going on in the brain when we're completing a task, right? So you're not just 100% focused on this one sort of stimulus. So this is called the background connectivity. Everything that's kind of behind the scenes and that's going on in the brain. I'll talk more about these things, but the most important thing is that if I refer to the word functional connectivity in reference to my task model, I'm not talking about the umbrella term, I'm talking about this task-specific, task-dependent stimulus. So what is resting state connectivity? I did sort of mention this already, but I'll reiterate it. So resting state connectivity is understood through the acquisition of functional MRI scans. Like I said, no task is being performed. Subject could be asleep, could be at rest, could be under anesthetic, could be sort of maybe meditating. These are all resting states. And what this gives us is an understanding of the brain's activation signatures caused by spontaneous low-level fluctuations. And so what I mean by spontaneous low-level fluctuations is essentially the small, random, sort of spontaneous increases and decreases of blood flow across the brain. So you can kind of just think of it as, you know, people working in the background once someone's at rest. Now, background connectivity. This is conceptually similar to resting state connectivity. However, it's measuring these small random spontaneous fluctuations that are not due to the task being completed. So anything that's not specific to the task stimulus. As I say here, assesses the brain's behind-the-scenes activation signatures when a subject is completing a task. So this is going to be kind of the focus of the results of my research, and we're going to be comparing resting state connectivity with background connectivity to get a better understanding of the general structure. So one more thing I want to talk about in terms of these building block ideas is how MRI data is collected. So I like to think about it as a bread analogy. We can pretend that our brain is a bread loaf, fresh from a bakery that's just come out of the oven, and we can cut our bread loaf into as many slices as we want. Now, you could either take a piece of bread loaf and analyze it, if you so pleased, and this would be like an fMRI scan of our brain. So each bread loaf slice is going to be a different scan, but we can put all those slices back together and really get a good representation of what the whole bread loaf looked like to start with. And now, in terms of this data, we also have that the images are typically taken around every 0.72 seconds. This can change from study to study, but this is just on average kind of how it is. Resting state imaging typically lasts longer than task state imaging, so around 15 minutes. Again, this can vary, but this is just more specific to the project that I'll be talking to a little bit later. And from the resting state imaging, we analyze resting state connectivity. As I said previously, from the task state imaging, we analyze both functional and background connectivity, so task-dependent and the task-independent stimulus in the brain. Now, one more thing is, so we have these slices, but what does this actually look like? What does this data actually look like when we're analyzing it? One of the ways to think about it is to think of a matrix, which is also sort of an Excel spreadsheet in a way. You can analyze MRI data three-dimensionally, and there's four-dimensional structure as well, but for my research, I'm only focusing on the two-dimensional structure. So in this sort of Excel spreadsheet style, we have that our rows represent our time dimension. So each row corresponds to the consecutive scans taken every 0.72 seconds. Now, before I talk about the columns, I just want to give a little example about what the rows actually look like, because they're not as intuitive as I think one would think. So let's suppose that we have a subject in the scanner. Doesn't matter what they're doing. They're in the scanner for 14 minutes and 40 seconds. This is just a random number. So 14 minutes and 40 seconds equates to 864 seconds. We know that a scan is taken every 0.72 seconds, so we end up with 1,200 rows. So 864 divided by 0.72 is 1,200. So we'd have 1 to 1,200 rows. Now, our column dimension is, or our columns represent our space dimension. So every single column in our Excel spreadsheet sort of way of thinking about it is a different location in the brain. And then all of the entries are these bold values. Those are the blood oxygen level dependent values that I talked about earlier that fMRI scanners obtain. All right, so now that we've talked about all these different components, building blocks about MRI scans, MRI technology, and as well as functional connectivity, I know there's been a lot of terms thrown around, but I'm going to talk specifically about each of these terms and how they apply to my research specifically. So from this point forward, I'm going to be talking about this large project, and then I'll be diving deeper into one subject and all of their data that I'm specifically analyzing functional, resting state, background connectivity on. So the Human Connectome Project, it's a really amazing project that was funded by the National Institute of Health in the United States. It was really a state-of-the-art project because there are so many subjects. There's over 1,200 subjects just in one subset of the project that I'm going to be using. And this was a consortium between 3 universities. It was called the Wu-Min-Ox Consortium. So that consisted of Washington University, University of Minnesota, and Oxford University. Something really important to note about this project is that the individuals were healthy. Now, we have lots of MRI data available to us as academics, scientists, but typically speaking, these are MRI scans that are done on individuals who have some sort of reason to be referred to get an MRI done, which means that we can't necessarily say that they're coming from a healthy brain. So we don't want to make inferences about the general human connectome from subjects that have potentially different disease or injury or trauma applied to the brain. Now, another really important thing is that this data is all openly available. So one of the things that they really wanted with this Human Connectome project was that it speeds up the process of understanding how to map the brain's connectome in general. So academics from all over the world are able to access this data, which is really fantastic. It uses the most advanced imaging technology. And now the most important thing for my research specifically is that each subject within the Human Connectome project and this Lumen-Ox consortium performed both task sort of scenarios, 7 different task scenarios, and they were also measured while they were at rest in the fMRI scanner. So this is important because generally speaking, you have a study that's done on maybe 40 individuals, and the study is analyzing resting state connectivity. So all these subjects are just going to be in the scanner at rest for a certain amount of time. Analysis is done and results are sort of found. Now we also have different studies where maybe 50 subjects are completing one specific task or 2 specific tasks, and then results are found. In order to compare these two things, there's really a lot of error that comes into play because we know that individual brains have so much variation that if we want to say something about the background connectivity from this task group and the resting state connectivity from this other group, we don't have as strong of evidence as we would like to have as we do here because we're analyzing this task data and rest data for each individual subject. So there's less variation going on. Okay, so what I have here is the specific data from what I say on here, subject 100307. From this point forward, I'm going to say that this is Phyllis. So we have data from Phyllis in the fMRI scanner, both when they're at rest and while they're completing the motor task. So we can see, as I mentioned about the dimension of the data, that we have our columns representing the locations and our rows representing our time points. For task and resting state data, we have the same number of locations in the brain. Nothing changes with that. The only difference is that they're in the scanner for a different amount of time. So you can see for the resting state data, we have about 1,200 time points, and for the task data, it's shorter. We have only 284 scans. And of course, the entries that I have in these in the actual cells are just made up numbers, but these are going to be our blood oxygen level dependent values. Now, the motor task is one of the 7 tasks that I chose to do my analysis on because it's the simplest sort of task from the Human Connectome project. In this task, the subject was asked to either move their left hand, their right hand, their left foot, or their right foot, or their tongue. And so the reason that the simplest task was chosen is because we basically want to be able to remove all of the information that is caused by the task. So choosing the simplest task seemed like the best idea in terms of not having to necessarily account for super complex brain functions that are going on to then be able to analyze background connectivity. So I'll talk about that more a little bit later, but this is just a visualization of the data that I'm doing all my research actually on. Now, I do want to talk about, or the way I'm going to talk about these 2 data sets is first, I'm going to do the resting state data and then the task state data. But before I go into those 2 different analyses, I want to talk about something that is applied to both data sets, but at different points throughout the analysis process. So regions of interest are things that are typically applied to MRI data to do 2 things. The first is that it reduces our data dimension substantially. As you can see here, we had over 93,000 columns, which is a lot. If you have 3D data, you have maybe over even 900,000 columns. So you can basically just minimize that a lot by applying these anatomical mappings of the brain. Something else that this does is it basically is allowing you to incorporate biological information that we know about the different regions of the brain and how they function similarly. So the anatomical mapping that I chose had 114 regions of interest. Most anatomical mappings are applied to the left and right hemispheres, and you can see this in the picture here with these different parcellations. However, the white subcortical region in the center there typically doesn't actually have anatomical mappings done. However, the Human Connectome Project has a way of grouping the subcortical region into 19 structural regions. So in total, the 93,684 locations are going to be grouped and have been grouped in my research process down into 133 regions of interest to simplify the problem and to incorporate spatial correlation within the brain. And like I said, these grouping sort of processes happen for both the rest data and the task data, but just at different times. So now I'm talking specifically about the resting state analysis. As you can see in the top left corner here, this is the sort of data set that I already showed you from Phyllis being at rest. We have our 93,000 locations and 1,200 data points. Now we are applying this region of interest anatomical mapping. So we group all of these bold values together, and the details of this I'm not going to go into here. But there's a bunch of statistical processes that occur that allow us to group our 93,000 locations into 133 regions. Clearly, this simplifies our data a lot. We now only have we still have 1,200 rows and 133 regions. But essentially what we're doing now is we're looking at the strength of connectivity between each of these pairs of regions. So that's what we get this yellow, it's called our connectivity matrix from. Now, the correlation coefficient is something that basically just measures how strong of a relationship there are between two different variables or things. So this matrix is going to be 133 by 133 rows and columns, and each location inside of the matrix is going to tell us the strength of functional connectivity between whichever pair of region intersects that point. So you can see that on the diagonal, they're all dark, and that's because, of course, region 1 is 100% correlated with region 1 because it's identical. And then we have these slightly darker sort of orangey values. And the way that we actually end up with an activation signature is that we determine a sort of threshold value and say that any connectivity strength that's higher than this threshold value we consider to be statistically significant. And again, there's lots of different ways that people can choose to sort of decide on this threshold value. But for my research, the value that we found caused there to be 3 pairs of statistically significant regions. And this is just from the resting state data. So I'm not going to talk too much in depth about the specific regions, but essentially we have our 3 pairs. They're basically all part of these inferior parietal lobule locations. The things in brackets are the different networks within the brain that these regions are a part of. And something that's important to note is that the default mode network is a thing, or is a network of regions in the brain that has been studied lots, and it's basically shown to be present when subjects are at rest. And it is in fact true that these 3 pairs of regions are all part of the default mode network. So that's a good thing, it makes sense, and that's kind of what we're going to sort of end with for the resting state part. And now I'm going to talk about the task model and kind of bring it back and compare it after. So before I talk about the specific task model for my data set for Phyllis completing the motor task, I want to talk about the general modeling construct. So in general, we have a response variable. That is our observed data. That's our measurements, any type of observed data. So for us, it's those bold values. You could think about it as someone measuring someone's height. Those are going to be our observed response variables. And then we have our input variables. The best way to think of this is that they're variables that we think as scientists are going to have an impact on our response variable values. So if you maybe chose eye color as an input variable for height, it would give you probably not too good of a model because we probably don't think that eye color impacts height of someone that much. However, if you chose age as an input variable, it probably has a larger impact. Now, the way that we actually measure this impact is that we calculate these beta coefficients. And so I'm kind of talking about it from left to right on the equation. So y, x, and then beta. These are our values that are estimated by the model. And they, in fact, tell us which input variables we chose are important with respect to the response variable. So how important those values are. So beta would be very close to 0 or 0, say in the situation where x, so our input variable would be eye color, and our output would be height, because it's probably not going to make a big impact. Now, the final thing I want to talk about with the general modeling construct is our error term. Again, this is calculated after we've estimated our beta coefficients, and it's found by taking the response variables, subtracting the input variables times their coefficients. So you can see in the dashed box here. And what it does is it tells us the amount of variation in the observed data that was not explained by our input variables, right? So we want to minimize this value. We want to have a very accurate model is one where our data has been explained very well by our input variables. So now, in terms of the actual data that I did not collect it. The Human Connectome Project collected it. So we have our Y box here is that exact matrix that I already showed you. So I'm just going to go back here to this slide. So the Y matrix is the motor task Excel spreadsheet at the bottom here with 284 rows and 93,684 columns, okay? So this is our Y. Now I'm going to skip over and just briefly mention that the error term, again, is the same size as our Y. This is just going to be calculated after we have found our beta coefficients. And then we have our beta coefficients that are going to be estimated in the modeling process. For this research, the process that was gone through is called a Markov chain, Monte Carlo Markov chain. This isn't super important, but if it's of interest to you, that's what was used. Now, this X input variable sort of matrix, what is this? This is definitely the most confusing part about the research, but essentially what I like to think about it as is that we really want to make an educated guess as to what our response variables are going to look like. So how do we do that? Well, this is just going to blow up that X matrix here. We basically know two things. We know from biological research that the brain responds to stimulus in a certain way. We have a stimulus right at the bottom there. We have a dip in the bold response, our blood oxygen levels. We have a peak, and then again, it slowly decreases. So this is something that's been studied over time. We know what this function looks like. It's the hemodynamic response function. And we also know the exact structure of the task that was being completed. So Phyllis was given a cue for 3 seconds that either said, tongue, left hand, right hand, and then she was expected to do a movement for 12 seconds after the cue. So this kind of gives us an idea of how we can combine these two pieces of information to give us our input variable matrix that we hope accurately represents what the response variables should look like. And now don't worry too much about this picture here, but I just wanted to give an idea because you can see here on the bottom we have our 5 different stimuli, right? Left foot, left hand, right foot, right hand, tongue. And the darkest blue sort of rectangles, you can say, okay, nothing was shown on the screen, and then there would have been a cue, and then the dark blue portion would be when there was some sort of movement happening from that specific cue, and then goes back to a state of rest. So this is our best guess at what the task model input variables are actually going to look like. Since our error matrix calculates the difference between our response variables and our input variables times a coefficient, we ideally want this value to be only taking into account that background behind the scenes activation that's going on if we've accurately accounted for all of the task-dependent stimulus in this design matrix or input matrix. Okay, so that was a lot of sort of details, but the point here is that we have this model assumption that we've sort of taken into account that the input matrix adequately explains the fluctuations of bold values that are due to task-related neural activity. That's what I was just mentioning, right? We used all the information that we have biologically, empirically, to calculate this input matrix, and we're hoping that it's doing a good job at predicting our response variables. Now, once we've done that, we then estimate these beta coefficients, like I said, with this MCMC algorithm. Not that important. But then after we've estimated our beta coefficients, we now find our error matrix. Since we've assumed that this model adequately explains the task-dependent neural activity, our error matrix is technically, we would hope, is going to contain only our background connectivity, the background fluctuations that are going on in the brain that are not specifically dependent on the task that's being completed. So in order to analyze this background connectivity, we then apply the same procedure to this error matrix as was done to the resting state data. All right, so this looks familiar, hopefully. So now the only difference here is that in the top left corner, I now have a same dimension as my response variables, but the values inside here are going to be our sort of our residual values. So these are going to be what's left over when we take the input variables times the beta coefficients from the response variable values. And again, these are just made up numbers here, so don't put too much weight on that. Now, we do the same process of combining these into 133 regions of interest because we want to end up with something that we can compare to the resting state to see whether there's similarities or differences between resting state connectivity and background connectivity. So again, we end up with our new matrix, which has 133 regions, or columns, and 284 time points. Again, exactly the same as what was applied to the resting state data. We find our correlation matrix using this RV correlation coefficient, 133 columns, 133 rows. Now this part has not actually been completed, so that's why it says draft here. This is just an example of what we might get out of the sort of end result. But essentially, we have two situations. Either this connectivity matrix is going to look similar to the one that we had for the resting state data, or it's going to look different. Now, this idea of similarities and differences is going to be quantified by some different tolerance or threshold, but these really are the only two potential outcomes. So what does this actually mean? So this resulting background connectivity matrix could be similar to the one we found from resting connectivity. What this indicates is that the regions that interact with each other when a subject is not completing a task are still interacting with each other while tasks are being completed. And essentially, we can think of these interactions as kind of like a foundation. So they're like the building blocks of a home. So this is one potential result if that background connectivity matrix is similar enough to the resting state connectivity matrix. However, we could have a situation where this connectivity matrix actually shows a lot of differences to that of a subject at rest. This would indicate that the functional connections that are not task-specific display their own unique and spontaneous activation pattern, which really could mean that they're foundational to completing a task. So they only arise when a subject is completing a task, but they're also very different from what's occurring in the brain when they're just at rest. So these are the two potential situations that we're going to find upon that final connectivity matrix being completed. And then, really, what does this help? As I said in the first couple of slides, understanding this background connectivity allows for future statistical models to use this information and improve the model, because essentially as close as we can get to the reality of what our brain is doing in a model, the better that model is going to perform for any sort of prediction power or sort of disease diagnostics or anything like that. Additionally, the overall goal of a lot of neuroscientists is to map the human brain entirely, map this human connectome. And of course, understanding the background connectivity of our brain is really important to this mapping process because we are typically at rest or completing a task. Now, lastly, we have the medical experts who can always use any additional information on the way that our brain is functioning to improve different intervention strategies, to assess the impact of trauma or injury on the brain, and as well to improve disease prognosis and progression of evaluations for the brain. So as we can see that there really are many different medical experts, many different fields that all benefit greatly from understanding the background connectivity of our brain better. So thank you for attending this talk today.